First of all, thank you, and I'm apologizing. I think I brought the Michigan weather with me when I came down here. So I am part of the MSU Wheat Breeding and Genetics team, and in our program, we like to explore different approaches, and we like to explore different breeding strategies. So one of those is the mini bulk system that Dr. Olson developed, which combines the concepts of speed breeding, RGA, and bulk breeding. And obviously, we do genomic prediction as a routine evaluation or selection mechanism in our program, primarily for grain yield. Now, recently, we started exploring the use of phenomics. So that includes hyperspectral imaging, infrared thermal imaging, and the one that I'll be sharing with you is when we tried exploring using UAV-based RGB and multispectral imaging. Now, our goal is to see how we can integrate that into our breeding program that relies primarily on genomics. So I'm going to jump right ahead into the summary of the phenomic evaluation or the UAV flights and grain yield. So I have a total of 663 genotypes that were evaluated in 22 and 23. By the way, these genotypes are primarily different. There's only like 50 genotypes that are represented in both years, since this is an active breeding trial. So this is composed of primarily advanced breeding lines and preliminary breeding lines. So for the UAV flight, I just used one. So that is the flight closest to flight at maturity that is closest to harvesting. So I generated a total of 30 vegetation indices, and I employed recursive partitioning and regressive tree model to do feature selection. So I have three feature selection evaluations, one using just the 2022 trial, one using the 23, and using the blues across years. And norm NIR and GR-NDVI were selected in all those three evaluations that I did. So, of course, our goal is to predict grain yield. First, we did your usual predictability assessment using cross-validation within those trials, and again, as a third training set across years. I used the usual data split of 80-20, and the predictability was just assessed by the Pearson correlation between the predicted and the actual grain yield of those in the testing set. For the prediction models, I use 8, your usual Bayesian models from the BGLR, Random Forest, XGBoost, and RBLUP. And I use the same model to do my phenomic prediction, straight up changing the SNP matrix or the SNP data with my phenomic data, basically using the 30 VIs as predictors. Now, to start exploring UAV data integration, our base model was RBLUP, and we started adding individual VIs as fixed effect or multiple VIs as fixed effect. Now, to take into account the multi-collinearity existing in those VIs, in those 30 VIs, we dimensionally reduced it using PCA and see what would happen if we used the PC instead as a fixed effect. Now, since these VIs are essentially phenotypes as well, so we use them as secondary trait to do multi-trait genomic prediction using factor analytic model and random effects model with unstructured covariance matrix. And also I use the PCs as another set of secondary traits. Now, I did some forward prediction as well to see what would be the utility of these models and approaches. So I used the 2024 preliminary yield trial that has 206 genotypes. Now, I intentionally removed all the checks and all the genotypes that were present in 22 and 23. So for this, I look at certain metrics. Obviously, I look at the phenotypic and genetic correlation between the predicted and the actual grain yield, but I want to share with you our results on assessing the indirect selection accuracy in here. So we get that by multiplying the genetic correlation between the predicted and the actual grain yield with the square root of heritability of the predicted values. So I'm just going to jump right ahead with the results from our predictability assessment. Obviously, there is a difference in the predictability from the genomic prediction depending on the training set that we used. Primarily, where's the pointer? There you go. So majority of the prediction models that performed relatively well are from your Bayesian models, also a little bit here from Random Forest. RBLUP is slightly performing lesser than what we expected, although in phenomic prediction, it was actually our blob that performed best. And random forest and XGBoost somehow have poorer performance, if you will. Now, the higher predictability that you're seeing here is because the VIs are primarily moderately to highly correlated with grain yield. So that's driving the predictability higher. Now, starting with the data integration. So first, I started integrating all of those 14 VIs individually. And obviously, there's a difference on how the predictability is depending on the type of VI that you integrated. Some of the VIs even resulted in lower predictability compared to your base R blob genomic prediction ability. And also, it's still happening that there's a huge difference depending on the trial that you used as training set, which we're expecting it anyway. Now, this is interesting. Apparently, if you started increasing the number of VIs going into the model as fixed effect, it's really going down. Like regardless of the training set or where you do your predictability assessment, it's always going down. Now, another thing is we, again, use the PCs. So using just the first PC is already good enough as a fixed effect, and that's primarily because your PC usually explains around 60 to 80% of the variation that you're seeing among the genotypes. In multi-trait genomic prediction model, using factor analytic model using the dimensionally reduced data or using the PCs resulted in higher predictability over using your random effects model and using your individual VIs as fixed effect. So by the way, the VIs that I used here, for this one is NDRE and norm NIR, and for this one is NDRE, norm NIR, and CTVI. Now, I'm gonna talk about our indirect selection accuracy, or from here on out, I'm gonna be calling this prediction accuracy. So obviously there's, again, difference in the prediction accuracy in the genomic prediction model depending on the training set that you used. Now this negative value is because the predicted and the actual grain yield has a negative genetic correlation. And again, the formula for that has the genetic correlation in there. And if it's negative, it's gonna look like that. For phenomic prediction, obviously it performed a little bit better, especially with our Bayesian models, but our blob really has really good prediction accuracy here using phenomic data, which is really nice. Now, when we integrate UAV data or your VIs, you see a higher prediction accuracy compared to either just genomic prediction or just phenomic prediction, which is really encouraging. But then again, you see this one because, again, the predicted values when you use CI-red edge as fixed effect has negative correlation with the actual grain yield. But earlier in the predictability, we see that as you increase the number of VIs going into the model, you just see decrease predictability all throughout. And here, there's different patterns depending on the trial that we used as training set. Using the 2023 trial, you see a sudden increase using just 2 VIs and apparently reaching around 0.25 or 0.24 in here. And actually, this one is the highest overall prediction accuracy that we're seeing in any of these approaches. Now, using the 2022, it's low. It's basically reflecting what we have seen in the predictability assessment. However, if you use the combined trials as training set, the prediction accuracy appears to be similar. Like it's not affecting that. Now, when you use the dimensionally reduced data, it's actually the first 3 principal components that would result in highest or higher prediction accuracy instead of just using the first principal component as a fixed effect. For multi-trait genomic prediction, it holds true. Using the first 3 principal components, using the principal components with factor analytic model using the combined trial resulted in higher prediction accuracy. So with that, we see that there is a variation in predictability and prediction accuracy depending on the training set and the model that you used. Also, overall, UAV-derived information in tandem with genomic data, resulted in higher predictability and prediction accuracy over using genomic prediction alone or using phenomic prediction alone. And this is a recommendation that we're trying to pitch, that you might want to use dimensionally reduced UAV information from combined trials, because as you may have seen, there's a glaring difference between using 2022 trial or 2023 trial as training set. Either you use it as a fixed effect or a secondary trait, it doesn't matter. And obviously, the predictability assessment by cross-validation does not always translate in forward prediction. Next is we want to see now what those what are the predicted values, how are the genotypes ranked in the forward prediction using these different models? Because right now, all I've calculated is just a percentage, how the models work, but I haven't actually seen the rankings of those predicted values yet. So with that, I want to acknowledge the MSU Wheat Breeding and Genetics team, especially Dr. Olson and Amanda, the co-authors for this project. And I also want to recognize the RSGIS team, especially Bob and Ava, for helping us with the flight and processing the images up until orthomosaic. So with that, thank you again for the opportunity, and I'll try to answer any question you might have. All right, no question? Oh,
Jeff's got a question first. Go ahead, Jeff.
See if Seth and I had the same question. So, can you speak more to how do you see phenomic selection? You started your talk by saying you use genomic selection currently, and you're looking to see how phenomic selection could augment it or improve it. I mean, genomic selection makes a prediction without having to plant and measure the phenomic covariates on the candidate, whereas phenomic selection requires you to do that. Yeah. So in the context of the wheat breeding program, what are the situations where you'll use that potentially in your variety development pipeline? Yeah,
so there's actually two pools of thought that we're bouncing off with that. Either you use genomic prediction early in the generation, let's say in F4 or F5, and then you use now your phenomic prediction later, primarily when you're doing PYT or AYT. However, on the implementation of this, of course, that's why I presented the using 22 and 23 combined as a training set. So in a similar concept with using genomic prediction, using your previous years of data to train the model, kind of like using that as well to implement it. Now, when are we doing the combined phenomic and genomic prediction, if you will? That's primarily, I would say, around PYT, or if they're in the family or line head row stage. So yeah. So, but there's still some logistics that needs to be really thought of in here. But those are, I think, some of the options where we can do both genomic and phenomic prediction, or even multi-omic prediction in the program.
I'll first follow up on Dr. Inman's question here. Do you use genomic selection to pre-select material before you plant it, or do you genotype the material once it's planted?
We do genomic prediction after we, so basically, I don't know if I'm gonna answer it as both. I'm just gonna answer it by telling you what we do with how we do the genomic selection. So we do select at F4 stage. So in a really, in all of those like bulk plots, if you will. So once we selected the plants, then we're going to do the genomic selection. So basically it's either after selection or before planting, regardless of how you look at it.
Okay. So it sounds like you're still planting the plants
regardless. Yeah.
So when you're talking about phenomic selection, I understood that you have 1 flight and 30 VIs.
Yeah.
Right? So your dimensionality there is pretty low. I know you've got some more flights. I mean, do you think that you would gain some more power by including more of the flights?
Oh, definitely.
So that you have greater dimensionality of that phenotypic information?
Yeah, that is true. Especially probably we haven't captured some of the traits that would have been otherwise captured either at flowering stage or at late vegetative stage. Yeah, definitely adding more flights, adding the temporal aspect of this would definitely improve our phenomic prediction accuracy. Pretty sure of that.
Thank you, Jhon, for your question. Sorry, we're out of time.
Thank you.
Let me introduce the